I'm making a continuation of Gaucher disease. So the location and the mutation is going to be found in the GBA1 gene on chromosome 1, specifically on the Q arm. And this mutation type is going to be a missense for this particular variant. It's changing a spare gene to serine, and it's going to cause a misfold and a misshape in this protein, and it's going to cause loss of function and a bunch of other phenotypes that you can see on the right hand side, especially lipid imbalance. That's mainly the main thing seen that causes the enlarged organs. So for the gene model, as I said, it's going to be on the GBA gene, chromosome 1, Q21, and this is going to be a 39 residue peptide, and there's a total of 497 amino acids. As you can see, that's very long. So this picture is showing it shortened. It's showing the 13 exons, which this gene has. And as I said earlier, it's a missense mutation, and it causes that amino acid substitution in one of the crucial exons. There are different types of other variants that are caused by nonsense mutations and frame shifts, but the most common form of Gaucher disease is going to be caused by that missense that I have in bold. And as I said, these mutations cause a loss of function in the main acting protein, which also has enzymatic activity that breaks down all of the fats in the lysosomes. And so here is another gene model showing the DNA, how there's the promoter region, where there's the specific consensus sequences that go along with this gene, and the mutation actually occurs around exon 9. And so this is where you're going to see that alteration of the amino acid and how that trickles down from the DNA to the mRNA and the mature mRNA. And since it is an exon, it's not going to be spliced. And that's why it's going to cause that mutation and have an almost like incorrect RNA. And then that's going to cause the misfolding and the loss of function. As you can see in the gene expression and in the biology, it does not allow the lipids to break down within the lysosomes. And this causes the enlarged organ phenotype, especially type 1 Gaucher disease. And you can see how it causes a lot of stress within the system. And with gene therapy, the pros, it does target the root cause, which means potential for more long-term acting treatment, reduces the need for a lifelong enzyme replacement therapy, could improve the organ and tissue function. Cons, it's still experimental, it's expensive, and there could be immune responses, and there's always that risk of off-target genetic effects with gene editing, and you can't necessarily be very specific and pinpoint to a certain extent where you're going to either create a new insertion or a correction for